Hello, this is chapter 11, which is mutation and the source of genetic variation. Now, are mutations a bad thing in the genome? Well, maybe not. Maybe we need to have mutations to make new genes and different variations of alleles so that we can make different proteins to adapt to a changing environment. And so changing your DNA sequence to produce new phenotypes to become better is also a possibility, as well as becoming worse, of course, if you make a protein that doesn't adapt well to the environment. Which one happens depends on that environment. Now, mutations are fairly rare. They happen about 1 in every million bases or so. And the mutation rate, of course, is the number of events that produce mutated alleles per locus per generation. And what that means is how fast are you mutating. Here are a few examples of some diseases that are caused by various mutations. And as you can see, most of them, if you look up and down these numbers here, this is 10 to the minus 5, which is 1 in 100,000. This is 10 to the minus 6, which is 1 in a million, and so on and so on. So the average mutation rate is roughly about 1 in a million-ish. So there are various factors that influence mutation rate. One of which is if the gene is larger, then it tends to have higher mutation rates. Now this is fairly straightforward. What this means is if you've got a gene of, say, 1 million bases, statistically you're likely to get one base mutated. If you have a gene of 2 million bases, statistically you'll have 2 bases mutated. And so you have more mutations in larger genes, just because you've got more bases to mutate there. There's something called nucleotide repeats, and these are associated with higher mutation rates. And we'll talk about nucleotide repeats in just a second. And also CG base pairs, that's cytosine and guanine base pairs, they are more likely to mutate than AT base pairs. CGs, cytosines, are especially vulnerable to chemical change, and so these cytosines change more than an adenine and thymine. Environmental factors can also influence mutation rates, including various chemicals and, of course, radiation. Now, some pollutants are everywhere. There is some natural radiation, such as solar radiation, and some radiation can be due to human causes, such as nuclear power plants. So here's a diagram of the radiation that you might find in the environment. And if you'll notice, about 80% or so, it's pretty much everything from here all the way around to about here, that's roughly about 80%, is coming from natural exposure. This is radon, which naturally comes from the soil, cosmic from the sky, of course terrestrial from the dirt around you, and just internal radiation. It's only about 20-something% that is coming from human-made sources. There are also ionized molecules that can cause mutations in DNA. These are molecules that have charges to them and can attack your DNA bases directly. And of course, free radicals, in other words known as protons and hydroxide ions, can also cause mutations in DNA. Now, if you don't repair these mutations, and this affects a gene that is responsible for preventing cancer, then of course that gene won't work, and you can get cancer from that malfunction of that gene. That's why this radiation can cause cancer, is because it affects the genes, which then don't make the proteins that are responsible for normal cell division. Chemical mutagens can also change your DNA, and there are a couple of examples. This one is a base analog. What that means, it's a compound that is very similar to a normal DNA base. An example here is 5-bromouracil, otherwise known as 5-BU, and this is 5-bromouracil here, and as you see, it looks very similar to thymine, which is over here. Not much difference between the two. So what we can do is grow a cell in the presence of 5-bromouracil, and as that DNA gets replicated, it will take the place of thymine in the DNA during DNA replication. And let's see what happens. So here is a normal DNA strand. We're going to grow it in the presence of 5-BU, and you'll see that this thymine here gets changed to 5-bromouracil, which is right here. Well, when this double helix starts to replicate, then we have semi-conservative replication, as you know, and this strand goes over here to replicate as normal, and so the existing A puts against a T, and where we get no mutation. But if you go here, if you take this 5-bromouracil, and here, of course, on the left is the original strand, when the new strand is made against that, 5-bromouracil, although it replaces a T, it actually binds to a G, and that's the source of the mutation. And so when this 5-bromouracil replicates semi-conservatively, the new strand contains a G instead of an A. So of course when these replicate, we end up having a CG base pair instead of an AT base pair. So 5-bromouracil has the ability to change an AT to a CG. There is another chemical mutagen, and this is nitrous acid, a very nasty acid, and this changes a cytosine to a uracil, but it does this directly. And what happens here is cytosine on here, and you see cytosine contains an amine group just at the top. And here is uracil, and uracil looks very similar to cytosine, except it doesn't have that amine group. So what nitrous acid does is it removes that amine group. It is a deamination of cytosine to become uracil. So what happens? Similar kind of system before, but we're dealing with different bases. Here is a standard CG base pair. The cytosine is deaminated to become uracil. When we undergo semi-conservative replication, half is okay, but this other half with the new uracil in it, when it replicates, it will put an A against that uracil, because A is just the RNA version of T, right? So it's going to pair up with A on the new DNA strand. The old DNA strand still has a C and still pairs up with a G. So when this new one starts to replicate again, we end up with these A's and T's instead of the G's and C's. So to change a guanine cytosine base pair to an adenine thymine, we use nitrous acid, which is the conversion of cytosine to uracil by deamination. There are also some nasty chemicals that bind directly to your DNA. They're called intercalating agents, and that's just a big fancy word for saying it binds directly. If you do this, you're essentially distorting that double helix. You're changing the shape of the double helix, and sometimes that can affect DNA replication and therefore affect the genes that that mutation occurs in. Here are two types of chemicals. One is called acridine orange, which is this tricyclic compound here, and this one over here is called ethidium bromide. It actually has 4 cyclic compounds, and both of these actually insert into the DNA double helix. So if we have a double helix in here, they will slide right in there along with, say, the bases. They're going to slide right in the middle, in like that, and that will distort the DNA double helix. This frameshift mutation is a special kind of mutation whereby you upset the codons during replication. So let's start talking about these different types of mutations, including frameshift mutations.